Hello, and welcome to Breast Cancer Action's webinar, Environmental Exposures, Why Timing is Critical. Thank you so much for joining us today. My name is Saru Kaiser, and I'm the Program Manager here at Breast Cancer Action. Joining me on the webinar today, we have three wonderful presenters. The first is Louise Greenspan. She's a pediatric endocrinologist at Kaiser Permanente, a clinical professor of pediatrics at UC San Francisco, and co-author of the book, The New Puberty, How to Navigate Early Development in Today's Girls. We also have Juliana Deardorff, clinical psychologist, associate professor at UC Berkeley's School of Public Health, and also co-author of the book, The New Puberty, How to Navigate Early Development in Today's Girls. And last, Barbara Cohen, director of the Child Health and Development Studies, a project of the Public Health Institute. Before we get started, I have a few quick announcements. Breast Cancer Action doesn't take money from any corporation that profits from or contributes to the breast cancer epidemic. Our work is largely supported by individual donors. Please consider making a $25 donation today to support work like these educational webinars. Presentation of environmental exposures, why timing is critical, will last about 45 minutes. At any time during the webinar, you can ask a question by typing it into the question bar, and we'll save time at the end to answer as many questions as we can. We want you to get involved with Breast Cancer Action. This webinar is a great way to do that. Stay tuned for other ways that we'll mention later on in the webinar. I'm going to start just by going over our agenda today. Dr. Greenspan will discuss the connection between early puberty and breast cancer. She'll also talk about some of the factors influencing early puberty in girls, such as obesity and chemical exposure. Dr. Deardorff will continue talking about factors influencing early puberty, focusing on the intersection between chemicals and stress. She will also talk about how these factors impact communities differently. And Dr. Cohen will talk about her work looking at DDT exposure on multiple generations of women and the impact this has had on breast cancer risk. A little bit about Breast Cancer Action. We were founded in 1990 by a group of women living with breast cancer who realized the power of community. They founded Breast Cancer Action because they saw a need for a grassroots organization with a unique understanding of the political, economic, and social context of breast cancer. Our founders, only one of whom is still alive today, knew that their private medical crises were part of a larger public health emergency. They felt that the experiences of those dealing with breast cancer needed to be heard to address the crises. We are the only organization that addresses the breast cancer epidemic at the intersection of breast cancer, the environment, social justice, and feminism. And our mission is to achieve health justice for all women at risk of and living with breast cancer. We recognize how issues of health and social justice are really intertwined through the work that we do. That's why we made a clear commitment to social justice as a foundation of all of our work, and I want to walk you through a few key elements of that. The first is we know that the breast cancer epidemic impacts communities unequally, and this leads to unacceptable differences in who develops breast cancer and when it develops, who gets high quality and timely treatment, and who dies from breast cancer. We believe that in order to address and end the breast cancer epidemic, we must tackle the root causes of these health inequalities, which are often the result of a complex interplay of culture, power, economics, racism, and sexism. We also believe that there's really no single injustice that can be effectively addressed in isolation. We know that injustices in our society reinforce each other in many ways and at many levels. And last but not least, in our work for health justice, we strive to be an ally. As an ally, we use the power and privilege we hold as an organization to build solidarity with communities who currently or traditionally have had less access to power, information, and resources. So what is the work that we actually do? We consider ourselves a watchdog for the breast cancer movement. We educate, organize, and take action for systemic change in three distinct areas. Our work on screening, diagnosis, and treatment examines the data from a patient perspective and ensures that women have access to accurate, unbiased information that will allow them to make well-informed decisions. We work on issues including breast cancer screening, healthcare access, and drug and device approval. Our work to address the root causes of the breast cancer epidemic focuses on eliminating the involuntary exposures to hazardous and toxic chemicals present in all our daily lives that put us at risk for an increased risk of breast cancer. And our work around pink ribbon marketing and culture is challenging the pink-washing hypocrisy by educating the public about the ways that the pink ribbon marketing and culture harms women. We also encourage consumers to ask critical questions about pink ribbon promotions and offer meaningful action that can really make a difference. So I'm going to take a minute to introduce you to all three of our presenters. The first, Louise Greenspan, is a pediatric endocrinologist at Kaiser Permanente and a clinical professor of pediatrics at UCSF, focusing on puberty and obesity. She is also the co-author with Juliana Deerdorf of the book, The New Puberty, How to Navigate Early Development in Today's Girls. Juliana Deerdorf, a licensed clinical psychologist and researcher with expertise in pubertal development and adolescent health. She's on the faculty at UC Berkeley and is co-author with Louise Greenspan of the book, The New Puberty, How to Navigate Early Development in Today's Girls. And last, we have Barbara Cohen. She's the director of the Child Health and Development Studies at Public Health Institute. She directs research examining how pregnancy protects against breast cancer and influences other health problems in mothers and their children in order to identify natural protective mechanisms that can be used for prevention. She also investigates whether early life exposure to environmental chemicals during pregnancy affects obesity, immune function, reproductive health, cardiovascular disease, diabetes, and cancer in mothers and their children across the lifespan. She holds a doctorate in epidemiology, a master's degree in city and regional planning, a master's degree in public health planning, and a bachelor's degree in zoology, all from the University of California, Berkeley. Welcome to all three of our presenters. I'm now going to pass it over to our first presenter.
Thank you very much. It is a real pleasure today to be speaking to this group and to be talking about puberty as an example of a window of susceptibility. Saru, I think you're going to have to take control of the slides. So if you can go to the next slide. So we are talking about puberty Julie and I really want to make sure that we get through the hype and the media attention which has been paid to puberty and get to the facts and the science. And we are discussing puberty because it's an example of a time in development of great susceptibility to exposures due to the rapid growth and the development that the body, particularly the breast tissue, is going through. And we define this as a window of susceptibility. But we also want to talk a little bit about what we mean by environment. And we hope by the end of both my talk and Dr. Deerdorf's talk that you will understand that the environment means a lot more than just, for example, chemicals or what's in our water. Next slide. I'd like to start with some definitions about what puberty is, and there are a lot of misconceptions. Most people, when they hear about girls' puberty, think that we're talking about first period. And we're not talking about first period. That's actually a very late event in the course of puberty. The first steps in puberty in girls are usually what we call thelarchy, or the onset of breast development or breast budding, and pubarchy, which is the onset of breast development, followed 2 to 4 years later by menarche, or the first period, as a very late event hormonally. Puberty is not a single event, but it's a series of changes that take place over this 2 to 4 years, and the timing, the tempo, and the sequence of these events may vary. Some of the changes are not visible, for example, the hormonal secretion overnight, which starts at the beginning, and some are very visible, such as the rapid growth development or the breast development. Next slide. In the old days, we used to talk about puberty in the setting of a cutoff for age 8 for girls for any pubertal development. In other words, any breast development or pubic hair before age 8 was abnormal. And we thought about menstruation starting before age 10 as being abnormal. The context here is for boys, who we're not going to discuss as much today, but for boys, we thought of 9 as the cutoff as being very abnormal. In 1997, a landmark paper was published. The first author was Marcia Herman Giddens, and she and her colleagues described in a cross-sectional study looking at a large group of girls across the country that puberty may have been starting significantly earlier. One of the concerns is that the age that you are when you get your first period is significantly related to your lifetime risk for breast cancer. Specifically, onset before age 11 versus onset after age 14 is a much higher risk. So in this setting, next slide, the NIH, the National Institutes of Health, decided to fund a large set of studies, which were initially called the Breast Cancer and the Environment Research Centers, which were funded by the NIEHS, the National Institute of Environmental Health Sciences, and the NCI, or the National Cancer Institute, to provide a consortium of basic scientists, epidemiologists, and community advocates, and we each had our own project. And we were investigating the links between mammary gland development, which in humans we call breast development, and breast cancer. And our epidemiology focus was on puberty as a window of susceptibility. There were also a basic science focus. So an example might be that we might look at a chemical in a girl's urine and see the timing of puberty in girls who had high or low rates of that chemical. And in the meantime, some of our colleagues in the lab might be studying the effects of those chemicals on breast cancer cells or on mice, breast development and breast tumor development. We all work together. And I'm going to present to you some of our data. Next slide, which really confirmed what had been found in 1997 in the Herman Giddens group. What we found was that contrary to the prior thought that age 8 was really the early cutoff, when we found that by age 7, 10% of white girls, 15% of Hispanic girls, and 25% of black girls had breast development, with Asian girls behind at 2%. And this was looking at a group of 1,200 girls from New York City, Ohio, and the San Francisco Bay Area. And we started studying them at age 6 to 8, and then we followed them for 10 years, and we've just wrapped up recently. This was a racially diverse group, as you can see. Herman Giddens had only studied black and white girls, and we were able to look at Hispanic girls and Asian girls as well. We see that there are obvious ethnic differences, but we do not think these are genetic. We think that these may reflect socioeconomic and exposure factors, which goes back to what Saru talked about with social justice. So I want to go on to the next slide and show you some of our data. The blue straight line was the girls from the 1997, and down at the bottom is age, and on the left is the percent of girls who had breast development. The dashed lines are our study. So when you first look at this slide, you could see that all the girls in our study are starting puberty significantly earlier than the girls in the 1997 cohort, which was the blue line. If you look further, that green dashed line was the overweight girls. So we can see that overweight is a significant factor here, but it's not the whole factor. All the girls started puberty earlier in our cohorts when we analyzed the data in 2013 compared to the girls 16 years prior. Next slide. So when we look overall, we see the persistence of the ethnic differences, with African American girls having an average age of onset of breast development at age 8.8 years, Hispanic girls 9.3, and Asian and white girls at 9.7. And next slide. When we go to the pubic hair development, we see the similar persistence of the ethnic differences, with Asian girls starting last. Only 2% of them at age 7 had pubic hair development. 6% of white and Hispanic girls, and 20% of black girls. Again, we do not think that these are genetic differences. We think that these reflect differences in exposures, and we are going to be discussing with you in my talk and Dr. Deardorff what we mean by that. Just to put this in context, next slide. We are analyzing our menarche or first period data, but we're still looking in general at the most recent study, which was the NHANES data, most recent NHANES, and that found about a half-year difference between African Americans and non-Hispanic whites, with African Americans getting their first period at 12.06 years, Mexican Americans at 12.25, and non-Hispanic whites at about 12.5. So next slide. Why does this matter? Why do we care about puberty? Well, there are a lot of consequences for girls who go through puberty earlier. The psychosocial ones are pretty dramatic, and girls who start their periods or start puberty earlier have a higher rate of depression and anxiety, behavior problems, delinquency from school, substance use, specifically tobacco, alcohol, and marijuana. They're also more overweight. They also have an earlier age of sexual initiation as well as a higher pregnancy risk, and they have body image issues. And many of these outcomes are already more prevalent amongst African American and Latina youth, so we are exacerbating that difference. Next slide. But these consequences persist into adulthood. And as I've said before, the earlier you are at first period, the higher your risk of breast cancer. And that's probably the most significant concern, and obviously for this audience is very worrying. But there are other things that still persist into adulthood for girls that were early bloomers. Depression and substance use persist. There's an increase in other reproductive cancers, such as uterine or cervical cancer. And there's a higher risk of overweight, obesity, and poor cardiovascular health. In fact, early first period, early menarche, is associated with higher all-cause mortality. The earlier you are when you go through puberty, the higher your risk of early death from everything. Next slide. So we're trying to delve into the culprits. Why? And we think that the main culprits are obesity, toxic stress, and endocrine disrupting chemicals. So again, puberty is a window of susceptibility. And each of these three categories of risks contain exposures which may be really hard to study in isolation. We can't analyze any particular chemical or any particular family dynamic. We have to look at the links between these three. I'm going to delve into obesity shortly. I want to just highlight toxic stress, and by that we mean toxic levels of family violence or neighborhood violence or other issues with psychological stress that affect the neuroendocrine system, and Dr. Deardorff is going to highlight those further. And also the endocrine disrupting chemicals, or EDCs. An EDC is a chemical which acts like a hormone in the body and may mimic hormones to promote or antagonize various hormones depending on which chemical we're talking about. So if we go back to obesity, next slide, I want to remind you from this particular graph that obesity, defined by the girls in that green dashed dotted line, they are starting puberty earlier to the left of the girls who are not obese, which is the other line, and then the girls who went through puberty in the 90s. Puberty is a majority, obesity is the majority of the effect on early obesity, but we don't think it's the only thing. And how does obesity cause early puberty? If you go to the next slide, it's a busy slide, but I'm trying to show that it's very multifactorial. And the relationship follows, if you look at the top, there are endocrine disrupting chemicals which may be present in the in utero environment, and they may affect prenatal growth and energy balance. Maternal weight gain and gestational diabetes are also things that may affect a child in utero and may promote obesity. While children are growing, they may be exposed to excessive nutrition, but it's not as simple as energy in, energy out. There are actually endocrine disrupting chemicals which may promote obesity. We call them obesogens. And we also know that there are many other things that can promote obesity that are not caloric. For example, the microbiome, or what bacteria your intestines carry around. Those may in turn be affected by endocrine disrupting chemicals or other chemicals that act not through the endocrine system. That visceral fat then goes and promotes the rest of the organs in the body to behave differently. So they cause the ovaries to make androgens, or male hormones, which can cause early pubic hair. They also cause the adrenal glands to do the same. They affect the liver and decrease a hormone that carries around other hormones, and then thus increases the bioavailability of testosterone or estrogen. And then those fat cells themselves make a hormone which can increase testosterone levels. All of that can promote insulin resistance, and that combined can then lead to a higher availability of estrogen and testosterone. And that is how we know that obesity can promote earlier pubic hair development, early breast development, and even really early true puberty. If we go to the next slide, there's an overlay with obesity and the chemical exposures. And these categories of chemicals here are the ones that we think may be promoting early puberty. Some of these are obesogens. Some of them are chemicals which may mimic hormones in the body. Phthalates are plasticizers. We studied all of these exposures, by the way, in our BCIRC studies, Plasticizers, phthalates, are things like PVCs or vinyl floors, shower curtains, but they're also in cosmetics, nail polish, shampoos, and lotions. And we think about the age of the girls that we studied. Marketing is a very strong drive for young girls to use a lot more cosmetics and other chemical exposing agents than we think are healthy. So these girls are exposed to phthalates through a lot of those. PBDEs are also a very big concern. Cotinine is in cigarette smoke. We looked at triclosan and phenols in general. Triclosan and phenols are used a very wide range of products. Triclosan is a phenol which is used as an antibacterial agent, and it can be in toothpaste and in clothing and in hand soap, and it seems to be an estrogen mimicker. BPA is a well-known phenol, and that can be a hard plastic or in can linings. It was originally created as a weak estrogen, and we know that BPA has estrogenic activities. And the PFOAs are the flame retardants, and there's a recent wonderful article in the New York Times which went through some issues with chemical companies and what they do, as well as phytoestrogens. Our BCIRC's findings, if you go to the next slide, looked at a bunch of these chemicals, and I'm going to go through these quickly before I hand this off to Dr. Deardorff. Mary Wolf was a colleague of ours in New York, and she looked at the phthalates. She tested the chemicals in the urine when the girls were 6 to 8 years of age, and she found that some of the phthalates were actually associated with delayed pubic hair. And 4 out of the 10 phenols had an action on puberty, either accelerating or delaying the puberty. Gail Wyndham is one of our colleagues here in the Bay Area, and she assessed the organic pollutants that are persistent in the serum at one time point, and we found that those flame retardants and other POPs are associated with delayed breast development. And then Hong looked at asthma and allergies, because these themselves can be related to chemical exposures in general, and found that those were associated with younger pubic hair development, but there were ethnic differences with blacks being more affected. Go to the next slide. So these are examples of work that's come out of our BCIRC, but we're challenged here because there are, it's very hard to isolate one of the chemicals and say this particular chemical is the cause. We know very little about the synergistic effects of the chemicals, and we also don't know how stress or the neuroendocrine system or cortisol activation, which Julie's going to talk about, can potentiate these chemicals. In addition, our study started late. We did not look in utero, and the other speakers today are going to speak to that and why that's important. In addition, there are a few other long-term studies with good puberty measures. Ours is one of the few ones. So this is an area where much more research is needed because, as I outlined, some of the chemicals may be causing early puberty, some may be causing late. They're certainly not good for us, but we don't have a clear-cut answer to say that one particular chemical or one particular compound is responsible for early puberty. If you go to the next slide, I do want to give people some resources that I recommend to my patients and that we recommend. The Skin Deep app is actually an environmental working groups database, and it's about to be relaunched as a different app. It's a wonderful database where anybody in the public can, for free, look up any chemical or cleaning solution or anything and find out if there's anything dangerous in it. And then for girls, we think when it comes to early puberty that the American Girl books are really wonderful resources. And then obviously we're proud of our book, The New Puberty, which we tried to synthesize all this data and put it together. And with that, I will hand it off to Dr. Deardorff.
Thank you, Louise. That was a great introduction to some of the things that I'm going to talk about today. So building on the research that Louise described, one of the things that we were most interested in addition to the contribution of obesity to puberty, which has been very well established, and examining chemicals, that research is still in its infancy. There's a large body of research that has looked at the contribution of stress to girls' pubertal timing. And what's interesting is that most of this research had really stayed within the psychological journals and scientific journals that psychologists were visiting, and it hadn't made its way into mainstream medical literature until recently. And so I'm going to give you kind of an idea of where that research is right now, and I'm going to see if I can advance this slide or not. There we go. Oops, I went back. Sorry, can you put me back? Great, thank you. So let me give you an overview of the things I'm going to talk about today. First, I think it's important to understand that there are multiple levels of exposure. So not just various chemicals, endocrine disruptors, and other exposures that affect puberty, but that these occur at multiple levels, and I'll explain what I mean to affect our health. There's also a literature showing that both maternal and psychosocial factors influence puberty. that's important to understand. And as Louise was forecasting, we know very little about the intersection of chemicals and stress, and that's really where the field is heading right now, is trying to examine those issues. I'm going to talk as an illustration of some of these things about another study that I'm involved in addition to the one that Louise described. It's called Chamacos, and I want to thank the PI of that study, Brenda Eskenazi, who's a close collaborator of mine, who lent many of these slides to me. So let's see. Great. So this is what I was talking about when I said that that puberty and just health in general is determined by a number of factors. And I think this is common sense to most people, that it isn't just one individual thing that resides inside our body that promotes disease or promotes poor health, but we're affected by many things in our environment. So Louise talked a little bit about genes, and while we don't think that genes probably directly influence the trend in puberty starting early, we certainly know that genes play a role in when we go through puberty, because girls who have moms who get their period early tend to get their period at earlier ages. But what's probably more important is how genes intersect with a host of other factors to contribute to this trend toward puberty starting earlier. So things like nutrition and obesity and health. And other things we know in the home environment and the neighborhood environment that also influence some of those factors. So for instance, if you don't have availability to healthy food in your neighborhood, then you're much less likely to be able to engage in weight control and maybe even physical activity, particularly if you're living in an environment that's not safe. And so things like violence and poverty also have influences on health, but also on puberty. And I think that most people think about puberty as an individual event that occurs within the adolescent and don't understand or haven't been exposed to the idea that many factors at many levels actually determine a girl's pubertal timing. So some of the recent studies that we have engaged in, and also some of our colleagues, have shown that maternal factors, particularly during pregnancy and just after pregnancy, play a significant role in when daughters of those mothers experience first signs of puberty. In this first study, we actually didn't have data about breast and pubic hair development, but we looked at menstruation, and we used national data, a very large sample of mothers and girls, and found that mothers who entered pregnancy at higher weights and who gained excessively during their pregnancies were more likely to have daughters who had puberty, who got their periods earlier. And this goes back to the model that Louise presented about how in utero effects may change the hormonal milieu within which the baby is developing and gestating in. Gestational diabetes, another factor that seems to influence girls in utero, also predicted early breast development in our study in the daughters of those mothers. And finally, on a more positive note, there's some preliminary evidence that we garnered from our studies, but it's also supportive of other research in the field showing that breastfeeding may be protective against early breast development, and so in daughters. And what's nice about that is that's a very modifiable factor that is both good for the child and good for the mother potentially if it's something that someone can engage in. There is a large literature talking about the psychosocial environment for girls and their pubertal timing, particularly the early psychosocial development that a girl is growing up in when she is between 0 and 5 years old. We know there's rapid brain development during that time that continues actually through adolescence and into early adulthood. But there's something about the environment that triggers puberty to start early if it's highly conflictual, highly stressful, and unpredictable in ways that suggest that there might be unsafeness in the environment, if you will. So things like family stress, low socioeconomic status, early sexual abuse, neighborhood disadvantage have been shown to lead to earlier puberty in girls. Father absence is also something that has been documented widely in the literature. Girls who grow up without their biological father in the home have about twice the risk of getting their periods earlier. And that's been replicated in many studies. So what does this matter for cancer? We've talked a lot. I mean, it seems to have an importance for the developing adolescent, but why do we care about its links to cancer? And Louise already mentioned that going through puberty early puts you at risk for cancer later in life, but of course that's not a bright line, and there's probably things in between, trajectories, if you will, of behaviors that may influence whether a girl is at risk for cancers later in life. And so some of the ways we think behaviorally that early puberty might affect you or emotionally that might put you at risk are as follows. Girls who go through puberty early are at higher risk for substance use, and both tobacco and alcohol use, as Louise mentioned. And they are at risk for initiating substances earlier, and we know that girls who initiate substance use earlier tend to use more heavily at early ages and perhaps throughout the life course. And we know that this can put youth later on at risk for breast and lung cancer. Also, adolescent overweight. So overweight is a risk factor for earlier puberty, but it's also a consequence of early puberty. So it's kind of a vicious cycle in terms of weight gain that happens for these girls. And we know that overweight might put people at risk for other cancers and also might limit physical activity if the overweight is too extreme, which also isn't good for promoting positive health. Adolescent mental health has been linked to early puberty, things like depression and anxiety, and we also know that that has implications for survivor outcomes of cancer. We know very little about how that influences inflammation and reduced immune functioning over the life course, but that's an important area of study. So let me talk a little bit about our current study, which is called CHAMACOS, and that stands for the Center for the Health Assessment of Mothers and Children of Salinas. This study is taking place in the Salinas Valley in California, and it is a birth cohort study which set out to investigate the health effects of environmental exposure, specifically chemicals, in low-income Mexican-American children who are growing up in this area. What's important to know about Salinas is that it's often called the salad bowl of the nation, and this is for good reason, because much of the vegetables that go into our salads across the entire nation are produced in this region. And as a result, the workers in this area and people living in this area are exposed to very high levels of pesticides. In fact, less than 5% at last count of the agricultural production that was happening in this area was organic. So Brenda Eskenazi initially started this study where she recruited a cohort of pregnant mothers because she really was interested in the effect of this pesticide exposure on the offspring of mothers who were being exposed when their children were in utero. And at the time, back in 1999-2000, the team enrolled over 600 pregnant women from clinics that were serving farm workers. Now you can imagine this was a very disadvantaged population. They were predominantly Spanish-speaking. Most were born in Mexico. Over half had less than 5 years in the United States, so fairly recent immigrants. 96% were living within 200% of the poverty level. And so just to contextualize this, this is a very poor sample. And many had a 6th grade education or less. And a large proportion of these women had worked in agriculture during their pregnancy. And even if they didn't, the vast majority had other agricultural workers in the home. And to compound that, many of the people live near fields that are being sprayed. So the study itself, as I said, began when the women were pregnant, and it has since continued. There have been many, many visits where data have been collected, both in terms of biospecimen, saliva, urine, breast milk, and various things at the beginning of the study. And then the children, once they were born, were also studied at various time points, starting at birth, 6 months, 1 year, and so on until they were 12 years old. At that point, I joined the study because of my expertise in adolescence and interest in pubertal development. Similar to the study that Louise described before, Brenda and her colleagues had begun tracking pubertal development in these children starting at age 9. And so we have leading edge measures in clinic of girls' pubertal development during this time, all the way through to where we are now, which is age 14. And we are just finishing up our 14-year visits and about to start our 16-year visits. So we really have a wonderful snapshot of what's happening to these kids, these youth, longitudinally. We've been able to track their development in a number of different ways. We not only looked at pesticides, but a variety of chemical exposures over the life course, in addition to all of the kind of measures of adversity and toxic stress that I was describing before, starting very young, as early as in utero. And, you know, unfortunately, as expected, these kids have grown up with a lot of adversity. So maternal depression in the sample is very high, which reflects kind of what we see nationally for immigrant Mexican women. Also, housing density is high. There's a lot of people living in these homes. When we did home observations, rodents were observed or present. People report food insecurity, so not being sure where their next meal is going to come from, or stretching their dollars to feed their families, or missing meals. There was also a home observation of things like toys and books and various things in the homes, and many people had none of these at age 1 for their children, in addition to many other things that I'm not showing here. Obesity is high in the sample. It's very high, and it's increasing with age, and we continue to see this. Stops at age 9, but we continue to see this increase in our adolescent population. And just to give you a point of comparison, it's even higher than what we see nationally in Mexican Americans, which in the United States, that's what that dotted line is. So it's just really kind of overwhelming in this sample. So you can imagine we started tracking their puberty at age 9, and that was even too late. Given these high rates of obesity, many of the youth in our sample had started puberty long before age 9. So what does this mean for good health and poor health? I think, you know, the answer is somewhat evident, but what I really want to stress is that when you have this kind of overexposure in a certain community to endocrine disruptors, you know, be they pesticides or other chemicals that are present in this environment, when you think about what people are cleaning with in their homes, when you think about some of the cosmetics and the beauty products that people are using in these environments where they can't afford to buy things that might be chemical-free. And then you think about that in combination with the types of adversities that I was describing that really do kind of illustrate high levels of toxic stress, then the formula, the synergy of these things really does point the road to very poor health outcomes, including various cancer risks. And we hear anecdotal evidence all the time from the families in our study of rare cancers and various other things that the families are very concerned about. They're concerned about their chemical exposures, and they're concerned about what's happening in the communities as they see it. So advance to the next slide. We call this kind of double jeopardy, this exposure to chemicals and stress. And Rachel Morello-Frosch, we borrowed that term from Rachel Morello-Frosch in a kind of seminal article she wrote in 2012 where she aptly labeled this double jeopardy. But when you think about the obesity risk on top of all of this, it really is more like triple jeopardy. And there's also folks in the field right now who are looking at underlying genetic vulnerabilities. So, I mean, triple, quadruple, there's a lot of synergy here and intersection that's happening between these various risk factors. And we, as Louise said, we can't just look at them in an isolated fashion. And I think Breast Cancer Action is dedicated to trying to understand some of the intersectionality of these factors. So finally, along with the mission of Breast Cancer Action and our team of researchers, we're very concerned with the social justice issues that this brings up. Pubertal timing may just be an illustration of a health effect. In a sense, it's a very, very plastic barometer for the environment. It moves fairly easily when you think about other developmental time points. And so with exposures to certain things like overweight and other things, you can move pubertal timing, for better or for worse. Again, many factors are likely intersecting with this, adding up to create risk for poor health, including various cancers. And truly, there are major social justice issues when we know that the most vulnerable communities are being subjected at very high levels to the most harmful chemical and environmental exposures, and stress, to be perfectly honest, on top of that. I need to thank a number of people. This is complicated research, and it doesn't get done without many, many colleagues, some of whom aren't even listed here. We have an incredible field staff down in Salinas. We do a lot of community outreach and community feedback down there. The families and the staff are really critical to this mission, along with all the investigators listed here. And the NIH has funded us for years to continue this research, and the importance of longitudinal research like this cannot be overstated. So thank you very much. I'm going to turn it over to Barbara.
Hello, I'm happy to be with you today. We're going to move to an event that happens to women much later in life. Saru, I'm not able to, maybe I can do it. Can you do the next slide, Saru, for me? Thank you. So an outcome that happens a lot later than puberty breast cancer, first occurring in most women at least 30 to 40 years after puberty, is even harder to study when you want to talk about early life factors and changes in health. So we're going to have to turn the clock back even further to look at breast cancer as an outcome of exposures in early life. So this is a very common pesticide that was used in very extensively beginning in post-war, was not banned in the United States until 1972. And this is an actual ad that occurred in 1947 that very much shows how heavily DDT, which was a pesticide, was used in the United States and elsewhere. And while it is a historic substance, it is still present in environmental samples and in our bodies in various forms. But maybe more importantly, what I want to emphasize is that the women who are exposed to this at a very young age are still quite alive. These are women born between the period of roughly 1945 and the mid-60s, and who were young and in at risk pre-puberty during those times. And those are the women now at risk for breast cancer. So keep that in mind as we go along. So the next slide, please. Before we can complete our journey on this talk, we have to talk a little bit about some facts about DDT. It was an insecticide, but what was in the can was largely composed of PPDDT, which is the insecticide. And during manufacture, there was also a contamination of the insecticide with another component called OPDDT, no way to really eliminate it. Over time, the insecticide itself is turned into its metabolite called DDE, and our bodies can do this, the environment can do this, animals do this, and heavily store DDE in fat. And so when we talk about DDT, we are not talking about only one chemical. And these, by the way, are not the only three that are derivative of DDT, but these are the ones that we've measured and could study most in our cohort. Use began in 1945, peaked in the late 1950s and the early 1960s, and was banned in 72, but as I said, is still present with us. Next slide, please. So what does it take to, go one more, Saru, please. Thank you. One more, or back. We want, yes, that's the one we want. So what does it really take to study the impact of an early life exposure, either in utero or at a young age, and breast cancer that occurs more than 50 years later? Well, it's really tough, and it's hard to study puberty, which has between an 8 and a 12-year delay, but now we're talking about 50 years. So we got very, very lucky here at the Child Health and Development Studies. We were able to look at a group of women who were enrolled when they were pregnant during the years 1959 to 1967. Those were very high DDT use years. And more than 15,000 pregnant women enrolled in this study. They're originally members of the Kaiser Permanente Health Plan. And nearly 100% of the pregnancies that occurred during those years happily agreed to work with us. Next slide. I'm not sure if we're going back or forward, but we'll see. Back then, please. So this is why the timing of blood collection is very important. The red dot here is California in 1963. Actually, it's the Child Health and Development Studies, CHDS, in 1963. The other blue dots are breast cancer studies, and we're measuring DDT here. We're showing the amount of DDT we can see in a person's blood. The other blue dots are studies that were conducted after 1970, with the Long Island study being one of the latest ones in 1996. And you can see that by that time, the levels of chemicals that could be found in people's blood had declined. And it's actually interesting because some of the women who were in the California study in 1963 could easily have been in the Long Island study later. So while these chemicals are persistent and stay a long time, they don't stay forever. So now we can go to the next slide. So why the CHDS data is unique for understanding exposure is, first, our blood was collected when DDT was heavily in use. And we already know from national data that by 1976, many women no longer showed the insecticide in their blood or the contaminant. This is really, really important because it is the insecticide and the contaminant that we found to be risk factors for breast cancer in our study. So if the insecticide and contaminant forms are not measured near the time of exposure, they can't be measured as risk factors. So next slide now. So this summarizes what we've been able to do. The red arc on top shows the DDT years, and the blue arc on, the green arc on the bottom shows the years when the blood was sampled. And we show that when we sample blood from a pregnant woman, we're also sampling the blood that her child or daughter was exposed to in utero. Next slide, please. Here's why this is important. So in pregnancy in 1960, the mother's breasts are preparing to feed an infant. And there are many changes going on in those tissues, but they're not the same as the changes that are going on in the tissues of her fetus, the baby that will become her daughter, which are just forming. And then finally, within her fetus are all the reproductive cells that will ever exist, in the girl in particular, that will become and form the third generation, the grandchild of the woman who's pregnant now. So there is the potential for any environmental exposure to a pregnant woman to influence 3 generations all at once. Moreover, because the reproductive cells are potentially at risk, there's the possibility that a change in those cells could be also inherited by the 4th generation to come, and that those changes do not necessarily have to be mutations in the classic sense, but ways in which genes interpret their instructions and proceed in an orderly fashion to make us who we are and have us function in a normal way. So the studies in pregnancy are extremely efficient, and they offer a unique opportunity. They're very, very problematic in that you may have to follow a cohort for more than 50 years to be able to do work like this. And we're just fortunate to have this one study where we can do that. Next slide, please. So here's what we found in the mother's generation. These tall purple bars represent an increased risk of breast cancer that is associated with a high level of DDT in the blood of women when they were pregnant. And this is their own breast cancer risk, the mother's risk. And the takeaway message here is that the level of risk that was correlated with DDT is a function of when the mother was first exposed to the pesticide. So women who were exposed prior to puberty had higher risk associated with DDT exposure, but women who were never exposed prior to puberty showed no association. So this is, we believe, a fairly classic demonstration of the vulnerability of the woman determines the impact of the pesticide or the endocrine disruptor on her own risk. And here we also see that the highest risk for the mothers was in the women who were the youngest when DDT was first introduced, when they were either in the womb or in infancy. Next slide, please. Now we turn to the daughters generation, and like I said, this is a very unusual study because the daughters who were born in the 60s are now around age 50, and we were able to look at their breast cancer risk in relation to what was in their mother's blood when their mother was pregnant with them. And here we found that the daughters whose mothers had exposure to OPDDT, which was the contaminant in the DDT bottle, had a nearly fourfold increased risk of breast cancer. And that among these women with breast cancer, their tumors were more aggressive, more likely to express a protein called HER2, which, and this is concerning because of the aggressive nature of these tumors. So this, to our knowledge, is the first womb to breast cancer study ever conducted in the world. And because these data like these are rare, it will be very difficult to replicate. So we have to talk about what we can do about that. Next slide, please. The impact of these findings suggests we can help prevent breast cancer beginning before birth. Next slide, please. So how can we help families now, given what we know? Well, we suggest that we can choose to limit exposure to chemicals for pregnant women and children, especially ones that are stored in the body for a long time, ones that disrupt or mimic a hormone like estrogen, and also chemicals that are not essential. And one thing that's left off this slide that I really would like to emphasize, even though it's not there, is that all of us can choose to support changes in society that reduce the use of suspect chemicals. And that truly is something we all can do, either by how we purchase or how we vote or what we say and do in our communities. Next slide, please. So where do we go from here? Well, we believe that understanding very early life exposures can help us to understand, treat, and also prevent breast cancer. Next slide, please. Why understand the past? Well, there are new technologies now like metabolomics, which is a way of studying all of the chemicals that a person makes in response to an exposure that can be applied to biospecimens, that's blood samples and urine samples and saliva samples that we've collected now in 3 of the 4 available CHDS generations to find diagnostic and treatment targets. Our example is, can we find the cholesterol for breast cancer? Cholesterol is a biomarker for heart disease. We try to lower it. We know it is associated with risk. And we're looking for something like that that we can help fix and we can use to predict risk. And we can use to identify people who are at higher risk than others. Next slide, please. However, even though this talk is a lot about history, I want to leave you with a positive note. We really have no evidence that breast cancer is an inevitable consequence of early life environment. And until we do, there's no reason to stop searching for a way to interrupt what may have started a long time ago. Next slide, please. This cohort, the Child Health and Development Studies, actually needs help now to continue. We've existed for 56 years by receiving federal grant funding for research. However, government research dollars are not a steady source for cohort maintenance. Things like storing our blood and urine and saliva samples, outreach to our cohort to keep them involved, extending our study to new participants in the next generations. And we are looking for partners very actively now, just starting this year, to endow the study for future generations, both for research and the benefit of the public, and also for the future generations of scientists who can use it and get to the very difficult questions that have been posed by the first 2 speakers, how the environment interacts with stress, and how social justice plays out over multiple generations. Next slide, please. Just some thank yous. These are some of the CHDS staff, not all. Over 56 years, you can well imagine there were hundreds of people who contributed to this cohort. I don't have time to thank them all. Next slide, please. This is our Participant Advisory Council. Unique to us, I believe, in many cohort studies is that we actually ask multiple generation members to participate in the council who help us decide on the science, help us disseminate our findings, help us determine what questions to ask, and even help us analyze our data and write our papers. This is a wonderful experiment we started 5 years ago, and I thank these people who give multiple Saturdays and lots of time to the work of learning how to communicate with us as scientists and with the community at large. Next slide, please. This is thank you to the late second director of the Child Health and Development Studies, who helped protect the cohort, kept this study alive. Next, please. And the founder of the Child Health and Development Studies, who understood the importance of these long-term data and really completely understood, although he would be surprised at some of what we do, that things that happen in utero are extremely important for the health of mothers, fathers, and families across the life course, but also across time. Next slide, please. The necessary acknowledgments for every single family member, which is quite a few of them, who participated in this study over the years, all the CHDS staff, the original Kaiser Plan and UC Berkeley collaborators who started this study, collaborators at multiple academic institutions who use our data and advise us, the Public Health Institute, the current home for the Child Health and Development Studies, which has provided a sound and flexible contemporary home for us, the California Breast Cancer Research Program, through generous funding, has helped us keep going and funded the Wound to Breast Cancer study we just showed you, and the National Institutes of Health, multiple institutes, Child Health, Environmental Health, and the National Cancer Institute, and the Breast Cancer and Environment Research Program, all of whom over the years in many ways have contributed to this work. Thank you, and that's it.
Thank you, everyone, for your presentations. And I'm going to quickly talk about how you can get involved with Breast Cancer Action, and then we're going to open it up to your questions. You can sign up for our e-alerts and find out what's going on and keep up to date on the issues. You can join us on Facebook and Twitter to change the conversation about the breast cancer epidemic. You can tell your friends, your coworkers, your family about this webinar and how they can learn more. And last, again, you can donate to support our education and advocacy work. I want to give a really big thank you to Louise, Julie, and Barbara for their presentations today. And now I want to take some time, the last few minutes, to open it up to your questions. People have been typing in questions as we've gone along. And I want to take a moment. Please continue to ask any questions you might have as we answer those. So I'm just going to start. Someone asked if they can have access to these slides, or if these slides will be available. And we do record all of our webinars and make them available on our website. It'll be up probably early next week, so you can check back and look back at the recording if you would like to. Barbara, someone had a question. If you could talk a little bit more just about the diagram you had of the many generations, sort of the vulnerability by generations. If you could talk a little bit more about that. A listener was sort of interested in sort of piecing apart that exposure at the different generational levels.
Sure. If you can get back to it, it'd be great. But if not, all you have to do is kind of imagine the pregnant woman and her belly and what is happening at that time to her body. And so pregnancy, of course, is all about the successful birth of a child and then the ability of the mammary gland of the breast to feed that baby after pregnancy. And there are many, many changes that the mother's own breast is undergoing in preparation for the birth of the child, her ability to make milk, immune system antibodies that are moving into the fluid at the very beginning that the baby can get to, and eventually the ability to keep up with the demand that the infant is going to make requires huge changes in the structure of her breast, and it becomes functional, and then later on changes back again. So during this time, this woman has a great and dynamic process going on inside her own breast. And so this is a vulnerable period for the pregnant woman herself. We know that because of other studies. We know that the timing of first pregnancy influences the breast cancer risk of a mother, for example. So now we move to the fetus. So the fetus is started from this fertilized egg, and all of its tissues must form, and the tissue of the breast of the fetus is present in utero and formed in utero. And what has to happen in order for that tissue to be normal is that it needs to follow a normal developmental program, and it needs to be influenced by the kinds of hormones and growth factors that were meant to be there and in proper quantity. It's also possible that the environment of the mother, the amount of food she has and her own weight, provides a signal to the fetal breast about how much structure to give it, how the ducts eventually will have to develop in order for that fetus eventually to nurture her own child. And then also inside the fetus is the ovary. Let's talk about the girl for a minute, because it's also true, different for the boys, but the boys also have reproductive apparatus that is developed inside the fetus, inside the mother. Those reproductive cells, the eggs that are in these girl children that are being born, who are these fetuses during pregnancy, are developed and sitting there, and reasonably potentially vulnerable at this time to all of the things that are circulating in the mother, because many of those things cross the placenta to the fetus, and then the tissues of the fetus, both breast and ovaries, are potentially subjected to the same exposures that was to the mother, sometimes in different proportions, sometimes in different ways, because the placenta is a bit of a filter, but many of the environmental chemicals that we're interested in do cross right over, and many of them are also found in breast milk. In fact, breast milk is one of the ways that people monitor environmental exposures to children and in the prenatal period. I don't know if I've helped at all, but so there you have the reproductive cells, which then become the fourth generation. And if you have altered them in a way that is reproducible, and there are a number of ways that we're learning about that we didn't understand for many years, but we're learning more about them, like the way the DNA is packed in those cells, the way start-stop code reading could occur in those cells. If those are changed by something that happened, in 1960, then the great-grandchildren could be affected and could be passed on, even though the fourth generation itself is not directly exposed to the environment in 1960 in this particular case. So we're concerned about the fourth generation. There are animal studies that cause us to be concerned, but to do a human study of this type, we're talking about 60 years at minimum. Great. And we're there right now. So,
thank you, Barbara. There's one other question. I think this is maybe to Louise and Julie. Someone is asking, are there protective behaviors that can be undertaken that would impact sort of puberty? Either I'll leave it there.
So I can start with that, and then I'm sure Julie can fill in a little bit. In terms of I think that's sort of a double question, I think, in terms of causing puberty and then the effects of puberty. There may or may not be things that we can do to prevent early puberty. Certainly eating a healthy diet and maintaining an active lifestyle are part of the things that can control children's weight gain, which may in turn help delay puberty. But more specifically, and this is the reason we wrote the book, because we really wanted to arm parents. There's a lot more than I can get into in 30 seconds here, so I definitely encourage people to get the New Puberty. But there are really specific behaviors that parents can have with their children who are in early puberty. And those negative effects that we describe both in my talk and Julie's talk are not inevitable if parents are able to provide structuring and scaffolding and really promote healthy behaviors, both in terms of the diet still, but also screen time and the limit setting that needs to happen so that children are still appropriately handled. We think part of the problem is if you start puberty early, by the time you're 10, as a girl you may look like you're 16, and so you may have an 18-year-old boy hitting on you, and you need to be able to teach the children how to handle older kids assuming that these younger girls should be one of their peers and therefore being exposed to older adolescent normative behavior, which is not normal at a younger age. Julie, I think you may want to add something to that too.
Yeah, so if I agree with all of that. If I'm interpreting the question correctly, are there things we can do to help prevent earlier puberty is the way I read it, which may or may not be accurate. But Louise, you hit on obesity and physical activity because that's where we have the most evidence that pubertal timing shifts when girls are overweight. We know much less about chemical exposures and endocrine disruptors and how they get under the skin. to influence girls' pubertal timing. So in that case, Louise and I both advise that people just exercise caution. You know, not everybody has the resources to buy organic and to ensure that they don't have chemicals in their shampoos and their lotions. But if you do, and if you have the ability to replace some of the things in your house, then why not do it? You know, it might not only have effects for puberty and other aspects of development, but other issues as well, like neurodevelopment and things that have been more closely linked to some of these chemical exposures, particularly in early life. Finally, in terms of stress and toxic stress, to the extent possible, I mean, I talked about how people in disadvantaged circumstances are just overloaded with a lot of uncontrollable stressors. But if you're in an environment where you do have control and you can reduce your stress levels in your family, then that's just good psychosocially for families in general and for children's well-being. And if you do have a girl who's going through puberty earlier, it's certainly going to be better for her to be in a stable, consistent environment with a lot of warmth and low conflict.
Great. Thank you. Thank you all. And we are over time, and so I want to thank everyone for their thoughtful comments and questions. And if we didn't answer your question today, you can follow up with us at info@bcaction.org. Also, you can watch your email for a short survey where you can provide some feedback on this webinar. Again, thank you so much, Louise, Julie, and Barbara, for your wonderful presentations, and I hope everyone has a wonderful rest of their day.
Thank you.
Welcome.